I also like to start that I find, for years I've been very much interested in alternative fragmentation technologies. And I think they are really necessary because I think we are already pretty good in doing tryptic digests. But as soon as it goes beyond that with other proteases or with glycopeptides or even phosphopeptides, it's really very useful to have alternative fragmentation techniques. And I'm really happy that SCIEX, but also some other vendors, are recently really investing again in getting methods around to get the alternative fragmentation methods going. They were always a bit slower, they were always a bit less efficient. But what I like most, and I say that already at the start about the new ZenoTOF, is really the possibility to do all these alternative fragmentation techniques with a relatively high speed, and also the efficiency of the fragmentation has increased as well. So that's where I actually want to talk about. But this is all in the context of the interest that we have in the lab for a couple of years now, and I will go through it quickly because I think most of you have seen this before, but we are highly interested in sequencing endogenous antibodies. And the problem there is that, this is extremely difficult. I try to explain you why, and I also try to explain you that we really need de novo sequencing for that. And then I also try to explain why we need novel mass spectrometry methods for that. So that's where I will go to. So basically what we do in my lab, we try to see which antibodies are in your blood or in other body fluids. There are antibodies of many different classes that we go after, the IgG1s, the IgA1s, the IgMs. And if we find the antibodies of interest, we also have to sequence them to identify them. And for that, we cannot use a genetic template because the antibodies are continuously maturing in the B cells. And therefore, the template is not available, so you really have to do de novo sequencing. So how many antibodies do we have? This is a slide I use a lot. And just to show you that the problem is so much bigger than what we call 20,000 gene problem, we can estimate from all the recombinations of the V, D, and J genes that a human person can make 10 to the 18th different antibody sequences. This is a staggering number if you compare that to 20 to the third number of genes. So all these different sequences are different antibodies, they target different antigens. And if we want to sequence antibodies, we cannot use a database of 10 to the 18 different sequences, so we have to do it this de novo. That's sort of the point I want to make. Now, it's also very problematic because every antibody is nearly identical in sequence. About 90% of the sequence is identical, and only the parts that are really variable are these CDR regions. There are 6 of them, 3 on the light chain, 3 on the heavy chain, and only there is this variation. So if you do normal trypsin digestion in the bottom up, it's very hard to get really enough peptides of these variable regions. You get a lot of more peptides from these constant regions. So you really is one of the problems in doing antibody sequencing. Now what we do now is we enrich antibodies from blood, for instance. We do that very simply with a tag that binds to the Fc part. Then we come in with a protease, we cleave off the FABs or the FAB2s, and the FAB is the top end, and that actually contains all these variable regions. So we make our life a little bit more simple. We don't sequence the part anymore that's on the bottom part, on the constant part. So, and I showed this before, if you do this in blood proteome and you look at the antibody profile, it actually turns out that our antibody profile is a bit more simpler than we thought. It's not 10 to the 18th. There are maybe thousands to maybe 10,000s to 100,000s of these clones available. They all have a different mass, and we can now measure these repertoires in a single patient by doing just LC-MS on this. And we get each mass represents a different clone in the repertoire, and all these masses are around 48 kilodalton, but we can really measure the retention time and the mass so accurately that we can really distinguish every FAB in this. And I'm actually quite happy, and I go over this quite quickly, that the chromatography of intact proteins was always rubbish, but nowadays with the new columns we can really separate all these FABs quite nicely and I think this is very important for our work. So I go over it. We have these clones now. Yeah, this is the plasma repertoire, but we can also do something else which I think is more relevant. We can use antigens to fish out only the antibodies that bind to specific antigens. Now here you see an example that we use as the antigen, a spike protein. And then you see that instead of this full profile that you see on the left, you see on the right the profile of antibodies that specifically bind to the spike. And there we still see that every human being has about 100 different antibodies that bind to the spike. And of course, these are the antibodies, if you're interested to develop antibodies against COVID, that you're interested in. So basically, our problem is not that we want to sequence all the antibodies on the left, we want to sequence all the antibodies on the right. So they are antigen-directed antibodies. But there are still hundreds of them, so it's still a polyclonal mixture, and still the sequences of all these antibodies are very much alike. So in my lab, we are developing and hope to develop new methods that can really help us to do high throughput antibody sequencing of these endogenous antibodies. Now, there are two approaches. Of course, one is the bottom-up approach and one is the top-down approach. And we actually have found out that you need really both, and you need the constraints from the top-down to really verify what the results are from your bottom-up approach. But I'm going to talk a little bit about this bottom-up approach. What do you typically do? If you have an antibody, and this is already done a lot when you have a recombinant antibody, you digest it and you use multiple proteases for that, and here are a few of the popular proteases mentioned. Then you generate a lot of peptides, and all these peptides need to be sequenced. And then you have tryptic peptides, you have chymotryptic peptides, you have peptides from the elastase digest. And now you have to find a way to align all these peptides with each other so that you get a full sequence coverage, at least of the CDR region. So that's sort of the state of the art at the moment. It's doable when you have a reasonably purified antibody, but it's way more difficult if you look at a polyclonal mixture, especially if this polyclonal mixture is very fast. Now, the other disadvantage is that you use here multiple proteases. They are expensive, it costs a lot of time, and you have to do all the data analysis. So the challenge that for this alignment and really getting all the peptides stitched together, we have solved with a software called Stitch, developed by Douwe here. And so this helps us really to align all these different peptides from the different proteases. But now the challenge was, can we do this more simple? And we teamed up with SCIEX actually, and also with Cinder Bio. And we said, can we not use one protease to get exactly the same as the 5, 6 proteases that we typically used? Now we said, we want to get rid of the multi-protease approach. Can we go to a single protease? We want to get better spectral quality in the data that we get so that our reads become way more unambiguous. And for that, we want to combine CID with EAD and also combinations thereof. And also in the data analysis, we are not so much focused on unique peptides. We are focused on how often do we read the same book. And can we get a sequence read by multiple reads? And so that's like reading a book not once, but twice or 3 times, or maybe even 10 times, so that you really know what the book is about. So, as I said, we teamed up. Most of the work I showed is still on the older sister or brother of the 8600. Most of our work is done on the 7600. Matthias already said what the advantages will be on the 8600. We were primarily quite excited about this because we had these alternative fragmentation techniques that we could combine. And also the efficiency of the electron-based induced dissociation in the xenotrap is really one of the best I have ever seen. So that made us very promising. So what I want to show, and it's slightly different from what Matthias showed, the ZenoTOF with these alternative fragmentation techniques, you can easily get reads of peptides that are maybe 40, 50, I would even say 100 amino acids long. And what you see here, we made a special annotator program, a spectral annotator program, and we use this colorful annotation at the top, that we not only annotate when we see a BMY ion in 2 colors, but we also annotate if we see the complementary C and Z ion, and even if we see the A and X ion. So for every amino acid, you see multiple hooks, and if we see 6 hooks, that actually means that we have 6 evidences that that amino acid is really there. So I say this is really careful reading a sequence of a peptide. Now the nice thing of the alternative fragmentation techniques, you all know that with HCD, you get good peptides from the C terminus, good peptides from the N terminus, but in the middle you're sort of screwed. But if you use all these fragmentation techniques combined, you really get these full sequence reads like I show over here. Now then we teamed up with SynderBio, and I'm always interested in new proteases, and they are not so novel. They were first described, I think, in 1995, but reinvented by SynderBio. And these are proteases that are very special. They work at pH 2, and they are extremely fast. And maybe in the past we would say the downside is that they're actually not so specific. So you see a specificity annotated at the bottom. So they are highly aspecific as well. And we started to explore these proteases for our work. And here I just show the workflow. We wanted to mimic first a polyclonal mixture. Now, it was just 4 antibodies. We used these 2 new proteases called Krakatau and Vesuvius because they are coming from these hot pools. We digest for a very short time at 80 degrees. And also with these proteases, we don't use reduction and alkylation. We just go directly as they come. Now we compare that, of course, with 2 gold standards, the trypsin and chymotrypsin, and we use the ZenoTOF for these readouts. Now, first of all, what we also did is we compared our CID with AID and the combination that in thermo language we would call ETHCD, but now we call it EACID. And basically what you see is that they gave us similar amounts of peptide reads. But of course, the difference is if we use AAD and EACID, we also get the complementary fragment ions and therefore better reads. Where that leads to, I just show you in the same peptides and analyze with HCD, with EAD and with EACID. And basically, maybe you cannot see it from the back, but you basically see again what I said before. With CID, of course, you only get the B and Y ions. And here it's actually already a pretty good reads, but you still have some gaps. But if you do this with these combined alternative fragmentation techniques, you see that you get a complete read of everything. And I'm really very happy with the quality of the data, the signal to noise. The annotations are really spot on, and there's almost no noise in these MS2 data. So for us, especially important is that we get then very long reads over these CDRs. And here you see just an example of one peptide. It covers the whole CDR, and we see amino acid coverage over every amino acid. But you also see below of this CDR, we actually get 5, 6 different peptides. And that was the great thing of these 2 proteases. The number of PSMs we get with every protease is about the same. But if we then look at the number of unique peptides, which is always a metric in our field, we see that because of the specificity of these 2 proteases, we get like 3 or 4 or even 5 times more peptides by using these 2 hot acidic proteases compared to these trypsin and chymotrypsin. If we also look at the charge distribution and the length distribution, we just see that these 2 proteases make all kind of peptides, short, but also very long, singly charged, but also charges up to 8 and 9. So we really see some peptides that are 10 kilodalton long, but we also see the normal, more tryptic-like peptides of 1 to 2 kilodalton long. Now we use this to see what is now the sequence coverage in peptides in the sequences. And you see in blue, you see trypsin and chymotrypsin, and then in, red and orange, you see the Vesuvius and the Krakatau. So basically, we get redundant peptide reads of the sequence. So that's what I said in the beginning. We are reading this sequence multiple times. We are reading this book multiple times. And because they overlay, you get multiple evidences of your sequences. Now, you get that especially very well if it's in a part that's still relatively constant. But even in the CDRs, we get not one read, we get multiple reads. And this really helps us to do very well. Now, basically, what you see that these 2 proteases outperform trypsin and chymotrypsin, but we also showed that they actually outperform if you would use subsequently trypsin, chymotrypsin, elastase, pepsin. So they do something special, they give many more peptide reads, and therefore I think are ideal for de novo sequencing. So we showed that this really leads to better coverage of the CDRs. Now the proof of the pudding is this is still matched to the sequences, so it's not truly de novo. It's just matched to the sequences that we knew of our polyclonal mixture. But the real proof of the pudding is, can we truly do it de novo? So we don't give the sequences of the 4 antibodies. We use for this Peak Studio, we use for this Stitch and Annotator, and we looked what is the performance of the SCIEX data for annotating those. And I was very happy that we can now, in this mixture, using the ZenoTOF, using the peaks, and the annotator and the stitch, we can really get confident identification of all these antibodies. For us, very important parameters is how many mistakes are we making. Now we see that the number of mistakes with the Vesuvius and Krakatau are way lower than when using trypsin or chymotrypsin. So in the end, and I see that I have to finish, the message is you need good quality data, you need redundant reads. These redundant reads also come from the multiple fragmentation techniques. And these two proteases, I think they are pretty cool. They are very different. Also that we don't reduce and alkylate anymore really helps us to get a completely different picture of the proteome. Now some metrics you see as well. The protease is very fast. It does 20 minutes instead of overnight. It gives you many more peptides and more of these redundant reads. And with that, I think I should end. We use this now for de novo sequencing. I had a few slides that we can now truly get de novo sequences of relevant antibodies from blood, but there is no time to go into that. I can discuss about that later. With that, I think I just end and thank you for your attention.